them, and so we hop between one and the other. So this is why you are on your own.
Hi, David.
Hi, hi.
Okay, well, so I am delighted to greet again David Unnerød, yes, who is an associated professor. You have to, if you did something wrong, if you did something new in the meantime, you have to tell me, David. Associated professor at KI, and he's also affiliated researcher at the Department of Clinical Sciences and Intervention Technologies at KI. And he works on the kidney functions and uses has been using clearing and expansion microscopy in his research for quite a long time. So he's going to tell us all about clearing and expansion today. Please, David.
Yes, great. Thanks
Thank
for
you.
the introduction. So I'm a postdoc at Karolinska, but I'm a guest professor now at the University of Cologne. So that's
Hey,
my current status.
well done.
So yeah, I'm going to give this overview of tissue clearing or optical clearing and tissue expansion. So as you will see in the presentation, these protocols are kind of related to each other chemically, so that's why it makes sense to sort of present them together. Yeah, and I have applied, so I spent, yeah, it's 12 years now, almost, applying these advanced sample preparation techniques in combination with different types of advanced imaging techniques to kidney research. So that's what I've been doing mostly. So the outline here will be just go through first tissue clearing, the basic concept of tissue clearing, and then present some different protocols that you can apply, and then give some sort of tips and tricks and starting points for your own projects. I will do the same for tissue expansion. And then in the end, I will also present a little bit of my own research, so how we are applying these protocols in our research. And by the way, anytime you feel that something is confusing, you can just interrupt me anytime and ask questions as we go. So, introduction to tissue clearing. So, as you all are probably aware of, biological tissue is non-transparent most of the times. So, what is the problem? Yeah, we have scattering inside the tissue. So, when we try to penetrate the tissue with an excitation laser, for instance, photons are scattered as we go deeper inside the tissue. So that means we are losing photons when we try to create this sort of focus down here inside the sample. The same thing, of course, happens to the emission photons or the fluorescence photons that we're trying to catch. They are also scattered. So this means that in a non-cleared biological sample, we can go, so here, this is a kidney sample. So at 10 micrometers of depth, so this is imaged with the confocal microscopy, so a low NA air objective. At 10 micrometers of depth, we can still see structures. 100 micrometers inside the sample, it's very noisy already, but we can see something. And 1 millimeter inside the sample, we are completely blind, so we have no contrast left. So why are photons scattered? Well, photons are scattered where there is sort of an abrupt change in refractive index. And this happens a lot inside a biological sample. So what we try to do with optical clearing is to homogenize the refractive index throughout our sample. So try and make sure that the whole sample that we're trying to image is of the same refractive index. And a little bit of history. So usually in most sort of tissue clearing reviews, there's a German guy called Spalteholz. He gets credit for inventing optical clearing. But there was actually a Swedish guy in Lund back in 1905. He started to play around with putting human embryos in different solvents. So this is a combination of, yeah, it's benzoyl and peppermint oil. So he managed to sort of make this human embryo transparent. Um, so maybe he didn't know it back then, but what he had done then is, of course, then to have homogenized the refractive index throughout the sample so that we could see through it. So how can we homogenize the refractive index in a biological sample? Well, lipids and proteins are of different refractive indices. So one way is to get rid of lipids. So typically we are more interested in the proteins, so we don't care too much about getting rid of the lipids. So that's one way of doing it. And then the mean refractive index of tissue is high. So that means that if we immerse the sample in water, then we will have a lot of refractive index differences due to this. So we can just immerse the sample in a solution with high refractive index, and this will also homogenize the refractive index. And there's also one other way, which is called hyperhydration. So there you try to sort of draw as much water as possible into your sample to, in this way, homogenize the refractive index. And all of the optical clearing protocols, they do one or a combination of these, of the above here. So you can group the tissue clearing protocols into kind of four basic groups. The first one being solvent-based tissue clearing. This is as it sounds like. So you used a clearing solvent. So what you have to do first then is to dehydrate your sample. And during this dehydration step, you then, of course, remove water, but you also, to some extent, remove lipids from your sample. And then you put your sample in a clearing solvent. So this is a solvent with high refractive index. And you leave it there for a while. This will also, to some extent, remove lipids. And also this high refractive index will then make your sample transparent. Then we have the simple immersion method. So this is the simplest way of clearing your sample. So this is basically just using an aqueous solution of high refractive index. And there are a ton of those. So this is pretty old now. This review is from 2015. And now, by the year, you have tens of new of these compounds added. So now there's a whole sea of different clearing solutions to choose from. And then again, hyperhydration. Mostly you use urea or urea-like molecules. And what you do then is to draw water molecules into the smallest compartments of your sample. And in this way, you can then make the sample more transparent. And then we have the last group, and that is hydrogel-based protocols. So the idea here is that you want to use a very strong detergent, so SDS mostly. And so you want to sort of remove all the lipids from your sample with this harsh detergent. So at least in the first original protocols, in order to keep the integrity of the sample, because this detergent is so harsh, you would then have to embed your sample in a hydrogel in order to link your proteins to the hydrogel to keep the integrity of the sample. So this, or sort of versions of these protocols, are the ones that I've been using in my research. So I will get back to this later. So for optical clearing, of course, as you know, the main or sort of the end goal is then to be able to image your sample volumetrically. So you don't have to section the sample, you can image the whole sample at once. So these images are acquired here in our lab at the light sheet facility that Stephen Edwards is running. And these are kidney samples. This is an adult kidney. So here we have TD tomato expression in the filtration units of the kidney, the glomeruli. And this is a late embryonic rat kidney, which is stained with a lectin, so it visualizes the tubules in the kidney. And these samples are both in the order of half a centimeter or so in size. So there are many protocols to choose from, as I already mentioned. So this can be a little tricky at first to know sort of which ones to choose. Uh, and things to consider there is, of course, what degree of transparency do I need? So usually, the more complex and the longer the protocol is, the more transparent your samples become, roughly. So if you don't need a huge amount of transparency so that your sample is pretty small, then you can start very simple. And I will get back to
Mhm.
this later. Um, in terms of labeling strategy, are you using fluorescent proteins, or do you have to stain your sample with antibodies? This is also something to consider. So some protocols are better for fluorescent proteins, some protocols are optimized for antibody staining. And of course, then also what imaging modalities do you have access to? And here I'm mostly referring to the refractive index. I'm sure you have already covered a lot of this in the course, how important it is to match your sample to the refractive index of the objective or the imaging system you are using. So yeah, all of these protocols, they have different sort of end refractive indices, and this is a very important thing to consider. So yeah, you want to find a protocol that works with your labeling, which renders the sample transparent enough to answer your scientific question, and which is compatible with the imaging method that you have access to. So again, I will dive into hydrogel-based optical clearing a little bit more, since this is what I've used in my research, and it's also a good sort of introduction to the tissue expansion protocols that I will talk about later. So the idea here of the original protocol was again that you want to make sure that all your proteins, the proteins that you are interested in, are sort of attached to this hydrogel that you want to introduce in order to be able to remove the lipids with this harsh detergent. So you fix your sample with PFA, and you have acrylamide monomers present here in green. So PFA will bind to the proteins, and then you will have the acrylamide binding to the PFA. And then you would polymerize your gel. Uh, so what you have done then is to very rigidly fix all your molecules to this acrylamide gel. And this then allows for dissolving all the lipids using this harsh SDS treatment without compromising the integrity of the sample. And as a last step here of this protocol, you then mount your sample in a high refractive index aqueous solution. So the last step here is always one of these simple immersion methods. Wait, let me just choose a laser pointer. So then I will talk a little bit about the protocol that we are using. So it's based on the Clarity protocol, but it's sort of highly simplified as compared to the first Clarity protocol. So the first Clarity protocol was extremely complex. They applied this electrophoretic tissue clearing, which required sort of this custom setup that you had to set up. So it was very over engineered, I would say. So the benefit of doing this was not huge, and it was just very complex to set up. So we have now optimized the protocol to be much simpler. And then a couple of years after the original Clarity protocol was published, then this protocol was published showing that you actually don't need, if you only do sort of uh, so if you skip this electrophoretic tissue clearing and only immerse your sample in a high temperature with shaking, so sort of passive clearing, as it's called, you don't need to introduce this hydrogel at all. So you can actually just fix your sample and then clear it in SDS. So based on this, we published our own protocol, which looks like this as a schematic overview. So it's basically just fixation of your sample. You then section your sample if you need to section it. You delipidate your sample in SDS from anywhere from 1 to 72 hours at 50 to 80 degrees. This depends on the sample size and sample type. And you then embed your sample prior to imaging in an aqueous high refractive index solution. So typically we use Histodense or Omnipaque for this. So this, I would say, is one of the simpler protocols, simpler clearing protocols. And it's reasonably fast. So it's anywhere from 5 hours up to 80 hours. And this depends again on the sample size and type. And also if you have to introduce, if you don't have fluorescent proteins and you have to stain your sample, this of course adds some time also. And I will get back to this later. So this you would do after the delipidation step and before embedding, you would add this antibody staining step here. Yeah, and then this is sort of some good starting points. So I would really stress that try to keep it as simple as possible. So these protocols are, they can be pretty sort of laborious to work with. So try to really start simple, and if this is not enough, then you can sort of work your way up in complexity. So for the aqueous protocols, I would say that this fast clarity protocol that we developed is a very simple starting point. It doesn't create the largest sort of transparency out of the protocol. So samples need to be fairly small. I mean, it works routinely for samples up to 1 millimeter, maybe a little bigger. But if your sample is bigger than this, then I would maybe consider some other protocols, such as Cubic is one that we had good experience with. When it comes to the solvent-based protocols, ethyl cinnamate is a very simple starting point. And this is completely non-toxic also, which is nice. So this is basically just dehydrating your sample, putting it in ethyl cinnamate, and then you have a transparent sample. So for the highest transparency, the Disco protocols, they create very transparent samples. They are a little more complex and also they have some toxic reagents. So this is something to consider there. But this was just to give you some kind of idea of where I think that you can start in terms of clearing. And then one slide about labeling cleared samples. So this is where you typically end up in trouble. So if you can work with samples with endogenous fluorescence, this is always preferable. Because then, of course, you don't have to introduce any probes into your large and thick samples. But then the clearing protocol has to be compatible. So some solvent-based protocols are not compatible. You can, of course, still stain for the fluorescent protein with an antibody, but then you're again back to the same problem that you have to introduce your probes. So this is something to consider. If you have to stain your sample with antibodies or dyes, Now, of course, these probes have to penetrate your thick sample by most often by just diffusion. And antibodies are large molecules, so they will diffuse pretty slowly into your sample. So this is something to consider. Then also, since you have a very big sample, you have a lot of epitopes for the antibody to bind to. That means that this large sample will consume a lot of antibody. So you need very, very high concentrations uh, in order for this to work. So long incubation times at 37 degrees, very high concentrations, and still it only works with very good antibodies. Because you have to crank up the concentration so much. So if your antibody is not of high affinity, you will just create a lot of background. Uh, so yeah, this is the step where you most of the time would fail, I would say, staining your samples. So that was everything about clearing. I don't know if you have any spontaneous questions before we move into tissue expansion. Any thoughts?
Or if anyone has already tried.
Okay, then we move into the expansion part. So I think most of you have heard about tissue expansion. So the idea here is very simple. Um, since the resolution of light microscope is limited, usually by diffraction, then can we circumvent it in any other way? Yes, so we can just swell the sample in space and we can move the molecules that we want to resolve from each other further away from each other. And then we have kind of solved this problem. So as I said, these protocols are all very related to these hydrogel-based clearing protocols in the way that you introduce almost always you introduce a hydrogel. And what is special about the hydrogel you would introduce with tissue expansion is that you introduce these sodium acrylate monomers. And sodium acrylate, when you polymerize sodium acrylate in a salt-rich environment, you will end up with these very curled up polymers like this. And if you then attach your biomolecules to this hydrogel and you move this sodium acrylate gel into a salt-free environment, so distilled water, these polymers will straighten up. So yeah, you have then moved all your molecules further away from each other, and your whole sample is now expanded. So you can image it at super resolution with a conventional microscope. So that's the whole idea. Okay, but in order for this to work, we have to solve one issue, and that is that you know that if you try to sort of pull a piece of meat apart, you know that you have some forces there inhibiting you to do so. So you have sort of internal forces inside cells and inside tissues that will inhibit expansion. So what you have to do then is to usually digest your samples. So you use very strong enzymes like proteinase in order to digest the sample to make it sort of, to allow for this expansion to happen. And the way they solved it in the first protocol, which is just called expansion microscopy, was to use these oligomer conjugated antibodies. So you would have these custom-made antibodies that are conjugated to an oligomer, which is then in turn conjugated to the fluorophore that you want to use. You then stain for your targets in the cell or in the tissue, and then you link this oligomer to your gel covalently. And in this way now you can very harshly just digest away your whole sample, and what you are left with is then a sort of fluorescent map of where your target used to be. And this gel you can just now freely expand in distilled water. So that was the basis of the first protocol. This had a drawback that you had to use these custom-made antibodies. So pretty soon after this, the Pro-EXM protocol was presented. So here, this allowed for working with conventional antibodies. So the way you would do it here is then to stain your sample with conventional antibodies, and then you would use a linker to link protein fragments to your gel. So to fix these proteins and link them to the gel. And then now you can digest, so you can start cutting these proteins here while these fragments are still stuck to the gel, so they will not be washed away. And yeah, in this way, you can then use conventional antibodies and achieve the same expansion factor as with the original protocol. Then there's a third solution called MAP, Magnified Analysis of the Proteome. Here you don't digest the proteins, but you denature the proteins instead. So this is also done with SDS at a very high temperature, which will then denature all your proteins and in this way allow them to expand more in space. So this original protocol also claimed to decrease the amount of cross-linking with PFA, because they claimed that the PFA cross-linking also itself inhibited expansion of the sample. So the original protocol used very high acrylamide concentrations. And in this way, every time a PFA molecule bound to an amine, it immediately bound to an acrylamide molecule instead of cross-linking to another protein. So that was the sort of original idea. So they showed that they achieved higher expansion with a high acrylamide concentration. But now there have been other protocols presented where they use a very low acrylamide concentration and they still achieve good expansion. So I think this is a little questionable. But the main idea here is that you denature your sample in SDS at very high temperature instead of digesting. And this then allows expansion and the same sort of expansion factor as these other protocols. Then there is a fourth solution. So this is actually hydrogel-free expansion. So it turns out that the tissue itself behaves like a hydrogel, meaning that it will shrink or swell depending on what solution you put it in. And then in this publication, they went through hundreds of different chemicals and just screened which rendered the sample most swelled. So in this protocol, you can see here that they managed to expand this brain around twofold or so, which is still a pretty decent resolution improvement. Of course, not as much as the other protocols, but with the advantage that here you don't need to introduce this gel at all. So yeah, there are now several protocols out there. And again, the question rises, which of these do I choose? So here I would say that, yeah, what degree of expansion do you need? So it's kind of the same here, that the more you want to expand your sample, the more complex the protocol, and also the more complex the imaging is of the sample. So just expand your sample as much as you need. And also here, labeling strategy is important. So some of these protocols are better with fluorescent proteins. Some are optimized for antibodies. And again, same question. what imaging modalities do you have access to. Uh, so yeah, if you have only have very low resolution imaging modalities available, maybe you have to expand your sample more. And then one other thing to consider here is that you are almost always dealing with water embedding. So, for instance, if you want to use a very high NA objective, like an oil objective, you will have a pretty severe mismatch in refractive index, and this can be an issue. So this is also something to consider. Yeah. So you want to find a protocol that works with your labeling.
So sorry, would you use then a water objective?
So I have actually this depends a little bit. So I it works, of course, to image these samples with an oil objective if you're close to the cover slip.
But you
But
are
then, of
never
course,
close to the cover slip since you're expanding them on.
exactly. I will get back to this later. So that's very, very hard to be close to the cover slip. And then it of course helps to use a water, even though the NA is a little lower. I mean, it's usually the only way to do it to be able to succeed. And you also typically have a little bit more of a working distance with those objectives than with an oil objective. Yeah, so then again, just some starting points. So this original protocol, I would basically not go there, but it requires these custom antibodies. So I would say no one uses this protocol anymore. Pro-EXM, it works with conventional antibodies, which is nice. And staining is typically carried out before you even start the expansion protocol. And for some stainings, this can be beneficial. So if you know that you have a very good staining in just, I don't know, normal immunohistochemistry, then maybe you want to work with this protocol. But for some antibodies and some stainings, it can be beneficial also to stain after the expansion protocol. So yeah, this depends a little bit on your target that you want to stain for. But it has been shown in several publications that you can actually increase the staining quality with, for instance, the MAP protocol, where you stain after expansion, uh, because you make the sample swelled and more porous, so it can sort of increase the accessibility of epitopes for antibodies. But for some antibodies, it doesn't work at all because you very strongly denature your sample. So this is always something to consider. I have not tried the cubic X protocol actually, but I have tried other ways of sort of slightly expanding your sample, just 30, 40, 50% or so. And this is, of course, nice because it's much simpler, less toxic, uh, and it's also easier to image these samples. So this is something to consider, that if you just need to push your resolution a little bit, maybe you don't want to go for this 5x expansion immediately. So here are some more comments about tissue expansion. So the protocols are often quite laborious and practically demanding, especially on the imaging side. So this is something that I think you should keep in mind. So one issue is that, as we already touched upon, you have this DI water embedding always. So RI matching, this can be an issue. And also, since these samples are very wobbly, so the more you expand them, the more sort of porous and wobbly they are. So it can be pretty hard to sort of mount them in a stable way. Uh, so drift can also be an issue. Um, And again, this we also touched upon already. So since you are now expanding, this is especially true for tissue samples. So at sort of interface or at the surface of the tissue, you will always have these valleys or unevenness. And these you will also expand a factor of 5. So now it can be pretty tricky to just sort of stay uh, close to the surface of the tissue. So this can really be an issue, especially if you're imaging something which is not that abundant in your sample that you have to look for. Yeah. So again, only apply if you really need this push in resolution to answer your question. And this is also something to consider that in my hands, since I have access to a super resolution microscope, this is usually the simpler option for me due to all of these things above here. That then my sample is not huge and expanded. I can mount it stably and nicely. So for me, it's just more efficient to image those samples. I should also mention that there are protocols that can expand your sample up to 20 times and even more. But then the same thing as above here applies, but yeah, a factor of 4 higher. So yeah, that's something to consider. So that was everything I had about expansion. So how much do I have until 3:25, or what was the?
Yes, 3:25.
Okay, good. So no questions so far?
I want to know if anyone had never heard of expansion microscopy. Can raise your hand. Right, interesting. Hmm. So it is a technique that
Yeah.
is already a little bit old. Yeah, right. But it's very useful. But again, you see the limitations now, so that's interesting. And if you have any questions, you can go
I
for it.
have a question.
Yeah.
Are there tissues that are known to be recalcitrant to this expansion, to be difficult to expand?
Yeah. So actually, the kidney is quite hard to expand, and that's due to there's a lot of collagen in some parts of the kidney. So very collagen-rich kidney can be hard to expand. So then you might have to use collagenase, for instance, to digest away the collagen. Other than that, I mean, I can just imagine that bone is very hard to expand if you don't treat it in some way, decalcify it or whatever you would do. Um, so yeah, but there are ways I think to deal with this issue for most types of tissue. You just have to use the sort of the right cocktail of enzymes to digest away these problematic parts of your tissue.
Okay.
So then I will just spend a little bit of time talking about. Oh, was there another question? No, I saw
Yes,
someone raise their.
I think so. Maria, you have a question?
Yes, sorry. Yeah, I have a question. I would like to know which is the best protocol that I can use for skin. I mean, clearing, yeah, the skin.
Clearing or clearing,
Yeah,
not
yeah,
expansion, right?
yeah,
Clearing.
clearing.
Skin, I cannot tell you from the top of my head actually. Uh, I don't have any experience actually of skin clearing,
I think it's
so
quite
I don't.
varied from one tissue to another. So maybe you'll have to have a look at the literature to find.
Yeah,
Yeah. Okay.
yeah. There are probably many. I'm sure there are protocols out there for clearing skin. So
Yes.
yeah,
I
yeah. There
think
should be something reported.
in the literature I have found that they use iDisco.
Yeah,
Yeah.
Mm,
exactly. So if iDisco works, I guess also this ethyl cinnamate could work, and that's a much sort of easier way to start at least.
okay. Yeah.
But yeah, I don't really dare to give you that much advice for skin.
Yeah.
Yeah.
Well, thank you. Yeah.
Yeah. Okay, so I'll just spend a few minutes talking about how I'm using these protocols. So we are looking at the kidney, and the kidney basically it filters blood, creates urine, and in this process it tunes the salt, sugar concentrations in the blood, also takes care of water excretion, etc. And the filtration of the blood, it takes place in the glomerulus. So these are schematically drawn in green out here in the cortex of the kidney. It's a tuft of very tiny capillaries. So you have the unfiltered blood entering, and then the filtered blood is exiting here, and in here is where the filtration takes place. And these capillaries are just a few micrometers in diameter, so the red blood cells will cover sort of the whole diameter of the capillaries. So they are really lined up in these capillaries. And on the surface, if you zoom in on the surface of the capillary, so this is like viewing a capillary from above, you find these tiny subcellular structures that are protruded by a cell type called podocytes. So this is when you view them from above, and this is a cross-section of the capillary wall. So there you have these finger-like foot processes here. As an epithelial layer, you have a basement membrane and endothelial cells here. So this is the blood side, this is the urine side. These are tiny, tiny structures. So historically, they could only be visualized with electron microscopy. So they are around 150 to 500 nanometers in width, these fingers, and the cell junction between them, the slit diaphragm, is around 50 nanometers. So only electron microscopy could visualize these structures before. So I started early on in my PhD work to look at ways with which we could resolve these structures also with a fluorescence microscope. So then this is Hans will give a presentation, I guess, later in the week about super resolution microscopy. So for the sake of time, I don't have time to present the super resolution microscopy, but you will hear about it. So super resolution microscopy, it increases the resolution of a fluorescence microscope. So it builds on a confocal microscope, but you can then increase the resolution a factor of 10 or so. So then the resolution should be enough to be able to visualize these structures with such a microscope. But what we found is that if we, with paraffin embedded samples, so this is like an immunohistochemistry that you would, like the gold standard method of preparing samples, we can start to see these structures when we are staining for proteins that localize to the cell junction between these fingers. But you can see that the background is quite high and the staining efficiency is not great. So what we then did was to add an optical clearing step. So just this SDS clearing step, and this allowed us to achieve this staining quality here. And this, I think, is an example of what I spoke about before, that also these clearing protocols, they slightly swell the sample and they remove lipids. So this makes the sample much more porous, and it's easier for antibodies to find their target. So that's probably the reason that we could achieve this in a cleared sample. So we published this in 2015, that it was only in a cleared sample when we applied STED that we could visualize these finger-like structures. And here we stain for the protein called podosin, which is expressed on both sides of this cell junction. So that's why you can see this sort of railway pattern here at some places. So this is resolving this 50 nanometer cell junction between the foot processes. So this was then the first time this could be done using an optical microscope instead of an electron microscope. And this had some advantages. So we can image samples in 3D, and we can sort of cover the whole capillary surface in this way. And by then developing quantitative tools or like semi-automatic segmentation tools, we could then extract quantitative parameters that describe pathologies in these structures. So that's one of the main advantages of using this as opposed to electron microscopy, that we can increase the throughput, and then we can drastically increase the sensitivity with which we can evaluate these pathological changes in these samples. So this back in 2020, we published a paper which then presented a new model for how kidney filtration works, and especially why kidney filtration fails in the disease state. And I'm not going into detail here for the sake of time, but the input data for all of this was the STED imaging. So this really sort of helped answering new questions in kidney physiology. What we have done a lot in the later years, so here we have then used deep learning to completely automate the analysis of these images. So we basically trained a deep learning network to, from such an image, segment these structures, and we could then in human samples, for instance, we can then very sort of sensitively quantify changes in patients. And this is just showing that, so with an electron microscope, you would catch maybe 10 or 50 of these structures, but here, just in one image, we can then automatically segment hundreds or thousands of food processes. So we can really sort of generate a lot of data with these new methods. And this is something we are now continuing to do. And I know that I think Robin is here in the audience. So he is now continuing this work and expanding it to visualize and segment other features of the kidney, and also combining this with multiplexing to sort of visualize the molecular landscape of the kidney filter. Then I just want to say also that we have applied tissue expansion. As I said, I don't work with it much anymore. So this was sort of a proof of concept paper that we published in 2018, where we showed that we can also do the same here with expansion microscopy and confocal microscope. So here we can expand the sample image with confocal and achieve the same results as with a STED microscope in a non-expanded sample. And we were actually the first ones to publish the combination of expansion microscopy with STED microscopy, but we didn't really dive much deeper into this. So we hid it just in the supplement of the paper, and then other people published it afterwards. Yeah. So I think that was everything I had actually. So just a little summary. As I already said, both clearing and expansion can be very demanding, and it's usually not a quick side project. So you really have to spend some time and put some thought into this for it to work. Again, start simple. I cannot stress this enough, I think. Like smallest possible sample, fewest amount of labels, because it's very easy to start from the beginning and think that, yeah, I'm going to clear a whole brain. I'm going to image, 5 targets at once, etc. But yeah, so start simple. That's my advice. And yeah, optical clearing and also expansion can sometimes be beneficial also for signal to noise ratio and labeling efficiency. This is something you can consider at least. But again, for some targets, it doesn't work at all. So this depends on the target. And seek help from people with lots of experience. That's also one of my advices. So with this, I would like to thank you, and you can send me an email anytime if you have some questions related to clearing or expansion or imaging in general. I'm happy to help.
Thank you, David. Thank you very much. So I would like to know if there are some questions.
Hi, I have a question.
Yes?
Oh,
Oh, you have some sort of echo.
are you muted?
Yeah.
Okay. I'm working with zebrafish embryos, and the tumor that I have is currently injected into the yolk sac region of the zebrafish embryo. So I'm wondering if your protocol works for the yolk sac region of the zebrafish embryo.
Oh, I know it works for the zebrafish embryo. The yolk sac region, that's too specific for me to answer. I don't know actually how, but I would assume so. I don't know. What
You're
are your
talking
own
about the
thoughts?
expansion that works, or you're talking about the
The
clearing?
clearing,
Clearing.
sorry.
Bit
Yes,
difficult
and
to
then
figure
also,
out because that's completely lipid. So if
Yeah.
you remove all the lipids, what happens?
yeah. But I mean, a zebrafish embryo is also in itself almost transparent, right?
Yeah, but it's just when you after you fix with PFA, the yolk sac kind of turns opaque.
Okay.
Yeah.
That I've never seen before. I also worked with embryos before,
Ah, okay,
but I
maybe
don't
that's
think it was opaque. But
maybe something is wrong from the protocol. Maybe I'll check.
but have a look if there is a protocol where there is a protocol of clearing for zebrafish embryos. You can try it and then see what happens to the yolk.
Yeah, okay, thank you.
Any
Oh,
more questions?
yeah, I have a very general question.
Yes.
For clearing or for expansion, is there like a upper limit of the size of the tissue you can do? Like you can do it in mouse organs, and is it possible to do human organ as well?
So they have done, I mean, they have done whole mice that I know.
Yeah.
And human organs, I don't know. I mean, I don't know what the world record is for, but you can make very, very large chunks of tissue transparent.
Okay.
Especially, I think it's the iDisco protocols or versions of the iDisco protocols. They have imaged huge samples, and then of course you need a custom microscope that can handle this, you know,
Yeah,
huge samples.
I'm just
So
curious.
yeah, yeah. So this, I don't know. It's very, very cool, but it's also, I don't know. Sometimes it's questionable how important it is to answer research
Yeah.
questions. You know.
With a very big, exactly. Yeah. Yeah. Stay
Yeah.
simple is a very good advice, I
Thank you.
think. Okay, but thank you very much, David. We'll see you again next year, hopefully.
Yes, sure. It's a
Um,
pleasure.
and